Hello, today we're going to be talking about recombinant DNA technology. And put simply, this is the ability to take a piece of DNA and move it from one location to another. So the objectives for today are for you to learn how to describe the major techniques used in manipulation of DNA and genetic engineering. And of course, genetic engineering often leads to genetically modified organisms, or GMOs, and we'll come back to that again later in the lecture. So we'll be focusing on several of the techniques which are used in molecular genetics today. We'll be talking in particular about restriction endonucleases. We'll also be talking about ligase, which you've come across before in this course. And then we'll be talking about plasmids as both cloning and expression vectors. But first, I'd like to give you an example of why we're interested in doing DNA genetic engineering. So, most of you will have heard of the disease diabetes. It's very common in today's society. People with diabetes have problems in regulating their blood sugar levels, and these problems involve the molecule insulin. So, first of all, let's have a look at a quick video which tells you what insulin does.
Hi, this is Mark from the MSOE Center for Biomolecular Modeling. The following animation will show the molecular mechanism whereby insulin triggers the uptake of glucose from your bloodstream. Soon after a meal, the digestive system begins to break down the food you have eaten. The carbohydrates in this food are broken down into a simple sugar called glucose. As this glucose is absorbed from your digestive system into your bloodstream, your blood sugar levels increase. Your circulatory system then carries the glucose to muscle cells throughout your body, where it is used to generate energy. And here's the important part. In order to get that glucose that's now in your bloodstream into your muscle cells, you need to have insulin present to trigger that uptake event. Insulin is a small protein hormone produced by your pancreas. As the concentration of glucose in your bloodstream rises, your pancreas senses this increase and is stimulated to release insulin into the bloodstream. The newly released insulin plays a key role in regulating the concentration of glucose in your blood, a process known as glucose homeostasis. The insulin that is now released into your bloodstream binds to the extracellular domain of receptor proteins found on the surface of liver, muscle, and fat cells. This binding triggers the autophosphorylation of the intracellular domains, which, in turn, phosphorylate a specific substrate signaling protein. This protein then phosphorylates other downhill signaling proteins, leading to an amplification of the signal at each step. This overall signaling process is known as a signal transduction cascade. One important consequence of this signal cascade is the movement of glucose transport proteins, called GLUTs, towards the cell surface. As these storage vesicles fuse with the cell membrane, the number of GLUTs present on the surface of the cells increase, allowing the glucose to enter the cell. As a result, the glucose concentration in the bloodstream decreases. The glucose is now inside the cell, where it can be metabolized to generate the energy in the form of ATP that is needed in all of your cells. And that is how insulin helps the sugar you eat turn into the muscle that you use.
So that's how insulin works. Now, in people with diabetes, they have problems with their insulin. And so one way to help someone who's a diabetic is to give them supplementary insulin. Originally, this insulin was derived by purifying it from animals such as pigs. However, we need to produce large amounts of insulin, and it was decided to take the human gene for insulin, put it into a cloning expression vector, a bacterial vector, and therefore produce large amounts of insulin which is easily purified to be able to treat people with diabetes. So now, let's go and look at the types of technology which is involved in being able to clone a gene from one place to express it in another. So what we're going to be looking at today is the origins of molecular genetics, and many of the processes that we're talking about come from bacterial and viral genetics. So fundamentally, what we want to be able to do is to cut a piece of DNA from one place and join it to another piece of DNA. And in this way, we're able to amplify that DNA sequence and express genes which are contained in the DNA fragment. I should point out that many of these tools are derived from natural systems which are found in viruses and bacterial systems. And so what we've done is taken something which happens in nature and harnessed that as a tool that we can use. So the first thing we want to look at is restriction endonucleases or restriction enzymes. So a group of researchers won the Nobel Prize in 1978 because they discovered that bacteria were able to produce enzymes which cut up DNA in specific sites. Since then, thousands of different restriction enzymes have been discovered, and over 600 of these are available commercially, which means that we can buy them and utilise them in a laboratory setting. Why are there so many restriction enzymes? Well, what they found is that different bacterial species contain different restriction enzymes. And so therefore, as many bacterial species as you can think of, there are probably that many restriction enzymes. So what is a restriction enzyme? What it is an enzyme which recognises a specific sequence within a piece of DNA. So if we look here at this blue piece of DNA, which includes this sequence reading from 5' to 3', AAGCTT. So that's on the top strand if we read from 5' to 3' on the bottom strand, AAGCTT. So what we say is that this sequence is palindromic. So for example, that's like the name Anna. So Anna is the same spelt forwards or backwards. So this 6-base pair nucleotide sequence is recognised by a restriction enzyme called HinD3. And what HinD3 does is recognise this sequence and then specifically cut the DNA backbone between these two A's on each strand. Now what that means is that if we break apart the hydrogen bonds between these spaces in the middle, we end up with 2 fragments of DNA. Each of those fragments has a 5' overhang of 4 nucleotides, TCGA, TCGA. But these 2 pieces of DNA are no longer joined together. So if we're able to do this in this dark blue piece of DNA, and then in this light blue piece of DNA, which has a similar sequence in it, we also digest that with the restriction enzyme HinD3, we will end up with similar sticky ends. Now we call these sticky ends because if you remember, A and T will pair, C and G will pair, G and C will pair, and T and A will pair. And this is by hydrogen bonding. So in this way, we'll be able to get this sticky end to recognise this sticky end and come together to rejoin to each other. Now back to one piece of DNA, but one different piece of DNA with half from one source and half from another. As I said, there are many different restriction enzymes. And so what we're looking at here is the 6 nucleotide recognition sequence for a different restriction enzyme called SAC2. In this case, the sequence is CCGCGG. Again, it's palindromic on the other strand. So in this case, the restriction enzyme will cleave here between this G and C and here between this C and G. So that means that our sticky ends will actually be 2 nucleotides in the middle, and in this case, we'll have a 3' sticky end, so a 3' overhang. Remember that many different restriction enzymes exist and they have independent sequence recognition sites. These recognition sites can be commonly 4 or 6 base pairs in length, although there are some restriction enzymes which have much larger recognition sequences. There are also restriction enzymes which will have a recognition sequence and then cleave right in the middle of that recognition sequence, and this will produce what we call blunt ends. So there's no overhanging nucleotides, so the 2 pieces of DNA are blunt. Those restriction enzymes which produce sticky ends are usually more useful in cloning though. Now if you remember, I said that restriction enzymes come naturally from bacteria. So why are those restriction enzymes there in bacteria? Well, this is one of the reasons. So this is a bacterial cell, which has been had attached to it a virus or a bacteriophage, which is then going to inject its DNA into the bacterial cell to try and replicate more copies of the phage. So here is the phage DNA entering the cell. Now, of course, the bacteria wants to protect itself from this phage. And so one way that it can do this is using restriction enzymes. So if this bacteria's restriction enzymes recognise sequences within the phage DNA and cut it into pieces, there'll be no replication of the phage, and therefore the bacteria is protected. So it's like an immune system for phage, against phage in bacteria, if you like. Now, those of you who are thinking ahead are probably wondering why these restriction enzymes don't attack the bacterial chromosome itself, the DNA of the bacteria. And there's a good reason for that. So every bacteria which has a restriction enzyme will have modifications in its own DNA so that this restriction enzyme no longer recognises the DNA. So here is an example of how the bacteria will modify its own DNA to protect it from its own restriction enzymes. So if we look at this C residue here, C nucleotide, if we add a methyl group to that C, shown here, this then becomes 5-methyl cytosine. Now 5-methyl cytosine still acts the same in terms of replication, so it still codes for a C, but because of this modification, it wouldn't be recognised by the restriction enzyme. So therefore, the bacteria can protect its own DNA and only have digested DNA from foreign sources. So we've talked about now how to cut pieces of DNA using restriction enzymes. So here we're showing that we've digested this DNA in orange with a restriction enzyme, which leaves this overhang here, CTAG. If we digest a second molecule with the same recognition sequence in it, with the same restriction enzyme, then we will get these overhangs shown here in yellow of GATC. If we put these two pieces of DNA together, the complementary sticky ends will align due to hydrogen bonding, and then we can use the enzyme ligase, DNA ligase, to repair the sugar phosphate backbone of the DNA. And what this means is now we have covalently linked these two pieces of DNA, and so therefore we have a recombinant DNA molecule. So in that example, we showed putting a piece of DNA into a plasmid. So what is a plasmid? Plasmids are independently replicating circular molecules, which are found commonly in bacteria. Now, they're not part of the chromosome of bacteria and not really part of the genome. They can move around independently between bacteria, and they often encode genes which are very important to the bacteria. So, for example, a plasmid which carries an antibiotic resistance gene will be of advantage to the bacteria, and so therefore will allow those bacteria to survive in the presence of an antibiotic, and therefore survive when the bacteria without that plasmid wouldn't survive. So here is an example of a plasmid. Now this particular plasmid is called PBR322, and it was one of the earliest cloning plasmids which was commonly used in a laboratory and is still commonly used now. So what we can see is that this plasmid is a circular piece of DNA which has an origin of replication. Now this is necessary so that this plasmid will be replicated by the bacterial systems which replicate the bacterial chromosome. So we have also, in this case, two antibiotic resistance genes, an antipenicillin resistance gene and a tetracycline resistance gene. And then what we can see here is that we have a whole lot of restriction enzyme recognition sites, including, if you look here, HinD3. Okay, so these are nucleotide sequences which are recognised by a suite of different restriction enzymes. And it's very important that we have these different restriction enzymes in order for this plasmid to be useful as a cloning vector. So what is important for a plasmid as a cloning vector? First of all, they're small size. So most plasmids which are used in cloning are about 3 to 5 kilobases in length. We want to keep them small so that they replicate easily, and they're also easily manageable within a laboratory setting. What we use them for is to capture, amplify and express foreign genes. So what we've done is we've taken naturally occurring plasmids, which occur in bacteria in nature, and we've adapted them so that we can utilise them for our experiments in a laboratory. And so cloning vectors have got all sorts of different features depending on what it is that you're requiring them for. But some of the commonly important ones are they'll have a selectable marker, which is an antibiotic resistance gene, so that if you take bacteria without that plasmid and plate them on plates which include ampicillin, for instance, those bacteria will not be able to grow and you'll get no bacterial colonies. Whereas if these bacteria have been transformed, that is, had the addition of this plasmid, which includes the ampicillin resistance gene, those bacteria will now grow on plates which have ampicillin, and therefore you'll be able to identify the bacteria which do have the plasmid inside them. Another important feature is multiple cloning sites. A multiple cloning site is a region of the cloning plasmid which includes many different restriction enzyme recognition sites. And the reason that we have this is so that we can utilise different restriction enzymes to clone specifically into that cloning vector. Another important feature is a colour selection system. So as well as being able to select bacteria which have the plasmid, we also want to be able to select bacteria which have the insert in the plasmid which we're interested in. And we'll talk about that a little bit more in a following slide. Another important thing is to have strong promoters. So if you remember, the promoter is responsible for how much of the messenger RNA is transcribed. And so therefore, if we want to express a protein at high levels, we need to make sure we have a strong promoter to drive expression of our particular gene. Another thing which is common in cloning vectors is to have plasmids which have high copy numbers. So plasmids can be found in low copy numbers, that is one copy of the plasmid per bacterial cell, or high copy numbers. The higher the copy numbers, the better expression we're going to get of the DNA and protein that we're interested in, and so therefore we'd like for them to be found in high copy numbers. So here we're looking further at that colour selection which I was talking about. So in this case we're talking about blue-white selection, which allows us to determine if we have an insert in the plasmid. So here's a schematic representation of our plasmid with an antibiotic resistance gene and also this gene called LAC-Z. What you'll note is that there's a restriction enzyme site in the middle of this LAC-Z gene, and often there's actually a multiple cloning site, i.e. many different restriction sites in the middle of this gene. And so what will happen is that in the plasmid, which has no insert, so this original plasmid itself, the LAC-Z gene will produce a blue colour when given a particular substrate called XGAL. However, when we cut with a restriction enzyme in the middle of this LAC-Z gene and clone into it another gene, or another DNA fragment, in this case insulin, into the middle of that LAC-Z gene, then we'll be disrupting the LAC-Z gene. It will become disabled and so therefore no longer function. So the protein, the LAC-Z enzyme, will no longer be produced, and therefore we will no longer produce a blue colour. And so if we pick the colonies which are white, we know that they have an insert into the plasmid and therefore are likely to contain the particular piece of DNA that we are interested in. Okay, so let's come back to our insulin example just to wrap up. So we've talked about plasmids and we've talked about how we can put a piece of recombinant DNA, so put in a piece of the insulin gene, for example, for expression into bacteria. Once that plasmid is in there and able to express the insulin protein, we can culture that in large culture vats and isolate our recombinant protein. So in the case of insulin, we would actually do this in two stages because the insulin protein is actually made up of two separate chains. There's the insulin A chain and the insulin B chain. So we would produce each of these, purify them, and then bring them together so that we were able to produce a recombinant insulin which is functional within a human. So if you know, what we're actually doing here is using a genetically modified organism. So we've genetically modified the bacterial plasmid, which is then put into a bacteria. So it's a different genetic material than was there originally. So this genetically modified organism is producing human insulin. That human insulin we then purify and give to humans to treat them for diabetes. I think this is an important thing to consider when we hear the debates about GMOs and whether we should have GMOs in our society. So just to review, the key concepts which we've covered in this lecture is that we wanted to understand some of the commonly used tools in molecular genetics. We've talked about restriction enzymes, we've talked about ligase, and we've talked about plasmids as cloning vectors. Now, as I describe these to you, hopefully they seem quite reasonable and simple and straightforward. However, remember that it was basic studies on the genetics of bacteria and viruses which uncovered the information that we required to lead to the discovery of the use of these tools. And it looks very clear in hindsight, but at the time the researchers involved had to work out some pretty complex situations.